Hello, my name is Mr. Chipman. This is 5.2 Meiosis and Genetic Diversity. Continuing on with Unit 5 Heredity, AP Bio. I also teach honors biology here at Murray High School in Murray, Kentucky. Let's talk about meiosis again. Remember, meiosis is the process in which haploid gametes are made by sexually reproducing organisms. As an aside, one of the middle school definitions of sexual reproduction is two parents. This is a terrible definition. Ask an oak tree if it's a good definition or not. The better definition of sexually reproducing organisms are organisms that produce gametes, right? And so if an organism produces gametes, it sexually reproduces. An oak tree, for instance, produces both male and female gametes. It can self-fertilize. So the idea of two parents is just bunk. It's like one of those easy things to say. All right, then, let's move on. Let's talk about, well, before we talk about nondisjunction, we're talking about genetic diversity. Sorry. Genetic diversity is created inherently in meiosis. It has to do with the not only the replication of DNA, but how DNA is sort of parsed out, how it's separated, what happens to that DNA during those parsing outs. Let's talk about nondisjunction then. So nondisjunction is a failure of chromosomes to separate. How can you know that? Well, let's look at the word nondisjunction. Junction means joining. Disjunction is not joining. Non-disjunction is failure to not join. What is the not joining? Separating, right? So failure to separate. We talked about two places where chromosomes separate. We mentioned in anaphase 1 that homologous pairs separate. And we mentioned in anaphase 2, sister chromatids separate. Non-disjunction can happen at either place. So you can have, let's look here. So up here, this is meiosis 1. Homologous pairs separate. In this particular thing, you have red, blue. You have two homologous pairs in this organism, right? And up here, red separates from blue, but notice in the bigger chromosomes, red and blue do not separate. What's that going to do? Well, blue is going to go over here to the right. Red, blue is going to go over here to the left. So now we have way too many chromosomes over here on the right. On the left, we have not enough chromosomes here on the left. And so our resulting gametes are normally one of these organisms should produce, each gamete should have two, right? Should have one of each. But that's not the case here. This isn't gamete, this is N for haploid, but it's N plus 1 because it has too many. This is an N plus 1. This is an N minus 1. It has too few, also an N minus 1. So when nondisjunction happens in meiosis 1, all 4 cells are going to be affected by that. You're going to have two N plus 1s, two N minus 1s. Here we go. Meiosis 1 happens normally. Homologous pairs separate, red to the left, blue to the right. We're going to look at red to the left, blue to the right in just a second, by the way. And in meiosis 2, when sister chromatids separate, one of them doesn't. And so here you have N plus 1, here you have N minus 1, and over here you just have N and N. So if nondisjunction happens in meiosis 2, half of the resulting gametes are not correct, right? Nondisjunction doesn't prohibit fertilization, so you can have fertilization with nondisjunction. You just have an individual that has too few or too many chromosomes. Trisomy 21 is a great example of this. Down syndrome. There are other examples of this as well. This shows what I was just talking about. You can look at this. Pause the video if you need to. I'm not going to explain it. Moving on. Crossing over is something else that happens in meiosis that causes genetic diversity. Remember, crossing over happens during prophase 1. Tetrads form. Chromosomes line up. This is my hands, but you get the idea. And when they do that, the homologous pieces of those chromosomes can actually sort of trade spots. Why is this not a problem? Well, blue and red have the same genes. They just have different versions of those genes. We call those versions of genes alleles. We're going to get into alleles in 5.3. Dad might have dominant A where mom has recessive A, right? A codes for eye color, but they just have different versions of that. And so if big A and little a swapped places here, it really wouldn't matter. That individual's still going to have eye color, but it's going to be with a different set of B's, right? A, B, C are not necessarily inherited together, and that is because our big A, big B, big C are not inherited together, but that's because of crossing over. We're going to talk about recombination frequency, which is one of my favorite things to talk about. Kidding. It's not. It's a pretty difficult concept. But you can see here that BC, now little a, is with big B, little c, right? Because the crossing over happened here. And you can see that. This is going to create genetic diversity. Again, genetic diversity is usually good for a population. Crossing over is a source of that. And this allows for a lot more kind of combinations that can occur in the genome. Crossing over is a normal process, by the way. I don't mean to make it sound like some sort of mutation. I've had students confused on that in the past, and this is a normal thing that occurs. And again, it's a good thing. Another way that this can occur is the principle of independent assortment. We're going to learn about Mendelian genetics soon enough, but this is one of Mendel's principles. And the independent assortment has the idea when there's multiple homologous pairs of chromosomes, those chromosomes assort themselves independent of one another. So we're going to zoom in. Here we have red, blue, and red, blue. We're going to call this big red, big blue, little red, little blue. So in this particular case, big blue goes left and big red goes right. Well, which direction does little blue go? Little blue doesn't have to go with big blue, right? It can make its own decisions. It can go with big red if it wants to. Now, if it goes with big red, then little red has to go with big blue. But they could also go together. The idea is that the big pair here sorts itself out independent of the little pair and vice versa. Or we could just call them A and B since that's what they're labeled here. Chromosome A here sorts itself out differently than B does. So you can have big A and big B together, but you could also have big A and little b together, right? So when we start looking at like possible genetic combinations, this concept of independent assortment is very important to understanding that. It's actually one of the things that my freshman students struggle with the most, but I don't have that as much in my AP classes. And then random fertilization. So when we use the word random is a word that gets way overused nowadays. So what does this mean? So female creates a bunch of gametes, male creates a bunch of gametes. What is the chance that gamete 1 is going to combine with gamete 2 over here, right? Well, it's a math problem. We're not going to do that sort of math in here. But the idea that gamete 1 has an equal chance to combine with any of these 4, right? It's random. Gamete 1 is not predisposed to combine with gamete 1 over here. There's no reason that gamete 1 and gamete 1 are supposed to match up, right? Or gamete 2 and gamete 2. It's random. Now, that being said, when we get to evolution, we're going to talk about how, there is some kind of fitness here. Sperm cells in particular have to move to their destination. And so some sperm cells are more fit than others, which that's this whole other thing. But as far as we're concerned, this should be a random process, right? And you've made a diagram before that shows this. This shows Punnett squares, which you've been doing since probably 5th grade. Punnett squares show that idea of random fertilization, right? You have one individual who is little g, and you have one other individual that is big G, little g, as far as their makeup, genetic makeup. And what is the chance that little g will go with big G? That is this idea. Punnett squares just show the concept of random fertilization. This is one gamete. This is another gamete down here. Leads to sexual differences, right? Which brings us the idea of the sexual versus asexual reproduction. Sexual reproduction, again, has to do with the production of gametes. There's a lot more diversity in sexual reproduction. Why? Random fertilization, independent assortment, crossing over, right? Those are why. Asexual reproduction is the production of a clone. Again, one parent, two parent are not really good definitions because they don't really tell you anything. And so what I prefer you understand is that asexual reproduction has to do with the production of a clone, whereas sexual reproduction has to do with the production of gametes. Those gametes are going to be genetically different than each other, which provides inherent genetic diversity, which is good. Let's look at a practice problem. Student observes that siblings from the same parents have very different combinations of traits, even though they share the same biological mother and father. Which of the following would be the most appropriate testable question to investigate the biological basis of this? It's good. I love these questions. You see 3 or 4 of them in the exam in May. How does this process of mitosis contribute to inheritance of identical traits? Again, this is not a good question because it's the wrong process. How does random fertilization ensure that each child inherits only maternal or, no, that's not the idea, right? Fertilization is both gametes coming together. What are the effects of environmental factors on the inheritance of acquired traits? This could be something, right? But we're going to look at B. We're going to look at D. How do independent assortment crossing over during meiosis contribute to genetic variation? Perfect question. C is just maybe not testable, or maybe there's too many things. So we're going to go with D because it is definitely something that can be tested, has been shown. I hope this is helpful. If it's not, let me know. If you have questions, let me know in the comments. Anytime, every time. I love your questions, and I love to help students be better at this class, which is a hard class. This is a hard subject. Unit 5 is one of those sneaky units. And so if you need help, please ask me a question in the comments down below. Thank you for watching.